Hey, everybody. We're going to get started for our weekly research rounds. It's my absolute pleasure that Han Kim is here to present his work for our research rounds today. Han has been a scientist at the Heart Institute since 2017. That's almost 10 years, Han. It's like it's getting there. He's a scientist here, as I say, as well as an associate professor with tenure in the Department of CMM in the Faculty of Medicine. The lab is the Functional Genetics and Metabolism Lab, and it's really an integrated research program that combines both sort of developmental biology, obviously cardiovascular function, as well as metabolism, which is really unique. So what he's going to share with us today is one aspect of the window of transcriptional mechanisms governing heart development, and in some of the I don't know today if that's what he's going to be talking about, the Iroquois homeobox transcription factors, as well as some electrophysiology, which is also very cool, as well as diet and metabolic intervention, specifically ketone body metabolism, actually regulates cardiac metabolism and homeostasis. His work he's highly published and accomplished in some of the leading reputable journals: PNAS, Nature, New England, Nature Communications. The list goes on. Han has been continuously funded since the day he arrived here at the Heart Institute, from the Heart and Stroke Foundation, multiple CIHRs, and actually one of the few investigators here at the Heart Institute who also has continuously held NSERC funding, which is fantastic. He's been awarded with awards such as the National New Investigator from the Heart and Stroke Foundation of Canada, ranked #1, the CCS Lilly Cardiometabolic Research Award, ranked #1. Probably this one too, Early Career Researcher Award from the Government of Ontario, and the Paul Dudley White International Scholar Award from the AHA. One of the other things that Han is well known for here at the Institute and has been recognized with the Mentor of the Year Award in Basic Science at the Faculty of Medicine is that leadership for his trainees. And for all those who have the pleasure of either being under his mentorship or working with the trainees under his mentorship, this is a really positive place to be, and where Han is really dedicated to fostering that next generation of scientists. So Han, please come and share your work with us. Thank you.
Thank you, Katie, for your kind introduction. And so also thank you all for coming today. So I'm pleased to share our research program and recent progress for over a few years, several years actually. It's kind of surprising that already been 10 years, almost. Yes. So my laboratory is a functional genetics and metabolism laboratory. So in this lab, the developmental genetics is the foundation of our research program. And we combine this with physiology and pathophysiology and metabolism, and also omics-based systems biology to address the key fundamental biomedical questions. So our research program has two main research pillars. One is to focus on transcriptional regulation in heart development and function and disease. And second pillar focus on metabolism, and particularly how it is regulated by dietary interventions and also energy substrate utilization, such as ketone bodies, and also the transcription factors. So I've been very fortunate to work with a group of talented and dedicated researchers and trainees in the lab. And here I put the current members in the lab, and really we had a really great time to address also scientifically, ask many interesting and we believe is important questions. And let me briefly introduce some questions that we've been tackling. And one question we have now is how the ventricular transmitter gradient is established and remodeled in healthy and diseased heart. So as you know, the cardiomyocyte across the ventricular wall is not homogeneous. And a prime example, classical example, is a T wave on the ECG, so which is made by the repolarization gradient within the cardiac muscle. So muscle cardiomyocyte in endomyocardium versus epimyocardium, they do have a distinct physiological characteristics. So how is it regulated? How is it established? And how is it affected? So that was our question. So in this study led by Julie Pan and Saif Debamne, at that time undergrad student, currently MD PhD in the UBC, and Yanao PhD student at the time. So we profile the transcriptome of endomyocardium, midmyocardium, and epimyocardium in both humans and mice. And we integrate these data and to understand what type of the transcriptomic profile is available on the muscle. And at the same time, we identify that IRX5 is transcription factor with the greatest diversity between endo and epi. And in addition, we found that IRX5 is essential to establish this gradient in healthy and diseased heart. Another question we had is why, in clinical condition, why congenital heart defect often co-occur with the extra-embryonic tissue defects, such as like placenta abnormalities. So that was our question. And this study led by the Termias Lenny PhD student with a team with Marwan and Lily, we identified that two transcription factor, IROQA and IROQA2, 1 and 2, especially in the cardiac origin, plays an important role in the vasculature development in extra-embryonic tissue. So in addition, our data suggests that there is common progenitor population that contribute both heart development and also the vasculature in the extra-embryonic tissue. So give some of the example that why this congenital heart defect can also accompany with extra-embryonic tissue defects. In the metabolism pillar, we've been interested in ketone body metabolism. And one of the drugs that we become interested in is SGLT2 inhibitor. And as you know, SGLT2 inhibitor is commonly used in heart failure patients as well as diabetes. And we tested that whether SGLT2 inhibitor can also treat the fatty liver disease. And this study led by the Sora Kwan in postdoc fellow and also the team with the undergrad student, they tested whether ketone, the SGLT2 inhibitor can improve. Indeed, as you can see in the liver histology, that the MPA glyphosine improved the fatty liver disease, while this effect depends on the hepatic ketogenesis. And our analysis also found a very interesting route that the effect of this SGLT2 inhibitor is actually not through the SGLT2 receptor, actually by some non-specific binding to other proteins such as HMGCS2 and then other kinase, the signaling pathway. In addition, we tested we've been working in the beginning that the role of intermittent fasting. We're trying to use this very common dietary intervention to treat the heart disease and any other disease. And one fundamental question we had is intermittent fasting equally beneficial for both males and females? So that was one question. And here we found that the study led by the Terme and then Shaza, previous master student, we found that actually intermittent fasting may play a little bit different role between male and female. In addition, we also found that this role is largely depends on the ketone bodies, especially through the immune cell involvement, especially the neutrophils. So this is a kind of example that we've been working on. But the fundamental and central question that connect that all our research program is how the molecular heterogeneity of the heart is established as a developmental biological perspective. And we've been interested in the cardiac conduction system. As you know, the cardiac conduction system is critical for the proper electrical stimulation and then also propagation of the heart. So so for the rest of the time, I'll share one published and two unpublished story about the cardiac conduction system. So first study is about that we uncovered the transcription regulation of postnatal cardiac conduction system heterogeneity, which was led by NRO, the recent PhD graduate and currently doing postdoc at Stanford, and also the great team of the Rimsha, Saif, and Mawan and Terme. So back to the cardiac conduction system background, as you know, every heartbeat is initiated by the electrical pulse generated in sino-atrial node. And this create a P wave and activate the atria first, and then it merge in AV node. There is some delay until the blood filling the ventricle, and then merge in the His bundle through the bundle branch and Purkinje fiber. They rapidly propagate and activate the ventricle in both left and right simultaneously, which create the QRS complex. So each region of conduction system, like SA node, AV node, His bundle, bundle branches, they're almost physically connected, but they play a very distinctive role. So how they actually function, those kind of things, and different electrophysiological phenotype. And understanding this electrophysiological phenotype in a molecular level is important because the defect in cardiac conduction system is directly connected to many diseases, including arrhythmia and also sudden cardiac death. So back to the history, and if you look at the conduction system development, I mean the histology and the finding, it is discovered more than 100 years ago by the several anatomists. And for example, is Dr. Sunao Tawara in early 1900, he confirmed the existence of AV node and also His bundle. And Dr. His found the His bundle, and also Dr. Purkinje identified the Purkinje fiber. So we use the same anatomical structure and definition of cardiac conduction system for our study and also the treatment. But in a molecular level, we still lack a lot of information. So the first question we had was how each CCS component in the postnatal heart molecularly different, like a SA node, AV node, and we also found AV node can be divided to the compact AV node and low nodal bundle, and also ventricular conduction system. We just call it ventricular conduction system, but the proximal bundle BCS, like His bundle and bundle branches, versus distal bundle branch of the VCS, the Purkinje fiber, how they are different. So that was our question. And next question is how this CCS heterogeneity is transcriptionally established. To address this question, we utilize some of the omics study, especially using single cell RNA sequencing combined with the spatial transcriptomic. So we use contactin 2 Rosati tomato, the conduction system fluorescence reporter mice. So we isolate the cardiomyocyte and backsort it, and then we perform the 10x sequencing, single cell RNA sequencing, and combine with GeoMax digital spatial profiler spatial transcriptomics. So by integrating this data, we led to the more accurate results. So first thing you show here, you see here is a UMAP, and some of you are familiar with it. So each dot represents a single cell. So we capture about 8,000 cells from the SA node all the way to Purkinje fiber. So we can bioinformatically separate them and what region they are. And combined with also the spatial transcriptomic, this is PCA plot showing, and it's interesting to see that starting with the SA node all the way to Purkinje fiber, you can see the linear alignment throughout the PCA plot, suggesting that they are very similar but also distinct. So by integrating this data, so we confirmed some of the known conduction system markers and also identify the novel CCS regional markers. And then so some of the example, I'm not going to go through everything, but some example is a GNAU1 in SA node, and then MPPED in the Purkinje fibers. Then we biologically validate these new markers using immunohistochemistry or immunofluorescence, or also the RNA in situ hybridization. And GNAU1 is expressed in SA node, and then some of the compact AV node but not in a lower bundle bundle. And then also MPPD is very specifically expressed in the ventricular conduction system in both His bundle branches, and Purkinje fibers. So we profile the conduction system markers and the genes. So the main question is, what established this heterogeneity? So we used a computational method called the CNIC, which is to infer the gene regulatory networks. So the idea is we're looking at each gene, each transcription factor, every transcription factor and every gene, and how they actually up and down regulate it. And by looking at the promoter region of each gene, we match how actually this transcription factor regulate downstream genes. So using this, we can identify the regulon, which is a kind of pair of the transcription factor and downstream genes. And we found that number of transcription factor and regulons are identified in a region-specific manner. So for example, the very well-known transcription factor SHOX2 is specifically enriched in a nodal cell, and then ETB1 is specific to the ventricular conduction system, and is also enriched in the ventricular conduction system. So this clearly showing that number of transcription factor express and working together to generate this molecular heterogeneity within the ventricular conduction system. So among these, we specifically focus on two transcription factors, the TBX3 and IDEX3. The reason is these transcription factor is very specifically expressed only in a cardiac conduction system, not in a contractile cardiomyocyte. And they expressed with an overlapping and unique pattern. For example, TBX3 is expressed in this nodal region, while TBX3 and IDEX3 expressed together in a His bundle and in proximal bundle branches. And while IDEX3 is specifically expressed in Purkinje fiber, suggesting they could have individual role and also overlapping role. So to examine whether these transcription factors may play a gene regulatory role, the safe looking at our marker genes in a more epigenetic landscapes. So we look at the ataxic data, which is the chromatin, open chromatin region data. So we look at the open chromatin region of atria or SA node, the ventricle, also the ChIP-seq data of TBX3 ChIP-seq data. We got it from the published data. So we align them to see whether the gene that we found is actually likely regulated by IDEX3 and TBX3. And for example, the VSNL1, you can see the SA node has a greater open chromatin and also aligned with a TBX3 chip, suggesting that VSNL1 could be regulated by TBX3. Also MPPD, but they have a very small open chromatin, but larger in the ventricle and also in the AB junctional region, also aligned with the TBX3 chip, suggesting TBX3 also can work on an MPPD. So this epigenetic data suggests that, okay, IDEX3 and TBX3 can be a regulator of this. So how can you also further validate it? So we established the in vitro model of CCS heterogeneity. So Yana isolate the neonatal cardiomyocyte from both atria and ventricle. And then she overexpressed the IDEX3 alone, TBX3 alone, and IDEX3 and TBX3 together. So what she wanted to achieve is recapitulating the structure, the cellular the properties. So for example, in a nodal cell where TBX3 positive, she overexpressed TBX3 in atrial cell, while in a proximal bundle branch, the VCS place where IDEX3 and TBX3 together, she overexpressed IDEX3 and TBX3 in the ventricular cell. And the distal VCS, she overexpressed IDEX3 in the ventricular cardiomyocyte. So we tested a lot of genes. So I give you some example. So VSNL1, which is specific marker in an SA node. And interestingly, when in atrial cardiomyocyte, when TBX3 is overexpressed, you can see that VSNL1 is largely increased. While IDEX3 and IDEX3 didn't do anything, but interestingly, when IDEX3 and TBX3 together, actually the enhancement is completely abolished. And more interesting thing is, all this finding is actually not shown in the ventricular cardiomyocytes. Means BSNL1 is specifically regulated by TBX3 only in atrial cardiomyocyte. So it's a cardiomyocyte context dependent manner. On the other hand, MPPD, which is a VCS specific marker, so nothing happened in atrial cardiomyocyte while it is increased by IDEX3 and TBX3 when this transcription factor is expressed. So this study is suggesting that same transcription factor in a context dependent play a very specific role. Means actually there are maybe atrial factor and ventricular factor together with IDEX3 and TBX3, the regulating the downstream expression. So in this first summary, we profile the molecular component of postnatal conduction system, and we found that CCS is not only made only the heterogeneous component that are different not only the anatomical and electrophysiologically, but also in a molecular level. And we also identified the gene regulatory network that establish the CCS component, and also found that using the IDEX3 and TBX3, we found these are probably some transcription factor. But as a proof of concept, we found that this combination of this transcription factor can establish the heterogeneity. So our finding is like really profile a lot of genes, but the main question we had ourselves is, okay, this is from the mouse work. And we use the mouse is a great tool to study many gene and disease. And indeed, a lot of cardiac conduction system development and transcription factor, our current knowledge is coming from the mouse work. But still, you can claim that mouse is mouse. And if you compare with the human, and this I trying to compare size of the heart and physiologically. So human heart is about 20 to 25 times bigger in terms of length, and heart rate is 10 times lower, and stroke volume is 1,000 times bigger. So when we compare the electrophysiological phenotype, they are clearly different. Means the key transcription factors are there, but there may be some downstream targets should be different. How they are different. And another thing is we use a lot of model organisms such as rat and zebrafish to study the heart. So is this the right way to study the human disease or human actually condition? What if the genes are different? So maybe some genes are available in a mouse, but not in a rat, or the other way around. So in that case, how can we actually identify what genes is good so that we can also properly choose the model organism. So to do so, we did a study that the title, The Conservation and Divergence of Transcriptional Heterogeneity in the Vertebrate Cardiac Conduction System. This study was led by the undergrad student, Mawan, and with the support with the Yana and then SAVE. So we, using our own data set as well as all the published data set, so the challenge is cardiac conduction system is about 0.1% of entire heart. So to capture the cardiac conduction system, you need a large data set. So which was actually challenging, but we actually got the data from the published data of the different stages, the developing the fetal heart, the 9 to 15 post-conception week or 19, and also 20 to 75-year-old, the human heart, and also the mouse embryonic heart and a postnatal heart, as well as rat adult, and also zebrafish embryonic and then adult zebrafish heart. So by integrating this, we're trying to capture, we did a comparative analysis to see what gene is the stage dependent and also species dependent. So this dendrogram shows how similar these genes are in a specific tissue, a development and species dependent manner. And interestingly, as you can appreciate that in the left side, all the dendrogram here is SA node. So all the SA node, regardless of species, they are clustered together, which makes sense. And the ventricular conduction system and Purkinje fiber His bundle is largely clustered on the right side. So overall, they are very similar, but how they are different in a specific manner. So first thing we compare is between mouse and human and developmental stages in adult. And I'm not going to go through every gene, but one example is GNAW1. I introduced the gene that in the essay note is both expressed in mouse embryo and human embryo, as well as human adult and mouse postnatal. And VSNL1 also consistently shown, and also MPPD we found in the ventricular conduction system in mice, also found in the human as well, suggesting that the gene that we also found can be seen in the human. And we also further using the spatial transcriptomic data of the human heart in an embryonic and adult heart. We're looking at these genes that we identify, GNAW1 and VSNL1, is enriched in the cardiac conduction system in human heart. In addition, we compare also the gene that we identify with the zebrafish and rat and see that which gene is actually consistently shown, but all zebrafish specific. So to further validate the gene that we identify, specifically the common gene in mouse and human, or actually mouse, the human and zebrafish, with a collaboration with Dr. Wenbin Liang, we using the human iPS drive cardiomyocyte. So we differentiate them to the ventricular cardiomyocyte-like cell, atrial cardiomyocyte-like cell, and also SA node-like cell. And we confirm the gene expression by when the ventricular marker is specifically highly expressed in ventricular-like cell, while atrial-like gene is expressed in atrial cell, and also actually SA node, because they are actually from the very similar the genetic background. And when we differentiate to SA node, SHOX2 and TBX18 is specifically expressed in SA node. Then we looking at the gene, the marker gene that we identify from our comparative analysis in this system, and we found that a lot of SA nodal markers such as PD1E and this contactin 4 and also GNAW1, they are specifically and like highly expressed in this SA nodal-like cell from the iPS drive cell, suggesting that indeed this marker that actually the functional role of this marker has not been tested. So we found that these are actually expressed. So next one is, okay, do we identify a number of genes? Are these actually meaningful? So what we meaningful in a functional level, or actually the disease level. So what we use is, we use the GWAS, genome-wide association study data, and we're looking at the variant in the GWAS, as well as we utilize a rare coding variant database. And we compare that whether the variant in this gene are associated with electrophysiological phenotype or heart disease. And here's some example. MPPD that we found in the ventricular conduction system is indeed, that it is significantly associated with the PR interval or ventricular arrhythmia or bundle branch block, suggesting that it is still correlation. However, it clearly showing the possible interaction and then role of MPPD that we found is related to the heart disease. So through the comparative CCS analysis, we reveal the both convergence and divergence of CCS component-specific transcript across species and developmental stage, which we hope to implicate this gene in the cardiac arrhythmia panel for drug targeting and also iPS reprogramming and regeneration. So to support the community, we also developed the interactive web application, the ccsatlas.com. So if you are interested in, this gene is expressed in cardiac conduction system, and so all the data that we presented here is implicated, deposited there, so you can test in this system. So the study that I'm going to share with you is, the title is the antagonistic balance between IRX3 and IRX4 in the ventricular non-compaction cardiomyopathy. So this study is being led by the Rimsha, the PhD student, with a team of many members. So the question that we had is, okay, so we identified the ventricular conduction system in a molecular level. And how the ventricular, so main question is how the ventricular conduction system is developed so that they can properly develop into mature ventricular conduction system. And ventricular conduction system indeed is developed from the trabecular cell, which is the progenitor population of developing heart, which is a mature cardiomyocyte. It's slightly different from compact cardiomyocyte. And this study from the Lucille McCorley and France, so she used connexin 40 reporter mice, and she found that indeed the trabecular cell can drive to ventricular conduction system as well as the contractile muscle, especially like a papillary muscles. So suggesting that this trabecular populate, the progenitor cell, have a two biphasic fate. It can be differentiated to BCS and or it can be differentiated to a contractile cell. So for us, which is interesting because of IDEX3 that we've been working on the Ottawa transcription factor family, and IDEX3 is specifically expressed in trabecular cell in developing heart, while after maturation, they are specifically expressed in ventricular conduction system, suggesting that in the trabecular population, IDEX3 is expressed, and then when the expression is maintained, they become BCS, but not the contractile cardiomyocyte. So the gene has to be turned off. Means there must be some kind of program that switching and deciding their fate decision of this trabecular progenitor cell. So what are those programs? So that was our fundamental questions. To achieve this, we using the publicly available data set. So which data set contained from the E9.5 all the way to postnatal day 9, the heart single cell data. And so we isolate the cardiomyocyte, and then we looking at the cell fate probability. So means based on the gene expression pattern, we can identify what is the origin of the cell and then how they actually differentiate to what type of cell. And they can differentiate to atrioventricular canal region or contractile cardiomyocyte or the ventricular conduction system. So this part is the origin of the cell. And especially when you're looking at this mid-phase cell, and then looking at the cell fate probability, they actually indeed have 50% chance to be the ventricular conduction cell or the contractile cardiomyocyte, means they actually biphasic cell. So we're looking at these cells more carefully and see what transcription factor is on and off during the differentiation. So these are the kind of number of transcription factors are only expressed in this early phase of ventricular conduction system. The phase changes or mid phase or late phases. And if you're looking at the IDEX3, and they are expressed, more this level in the trabecular region, and while they are differentiating to ventricular conduction system, this expression is increased a lot, which is consistent with our current knowledge. So interesting finding is another member of IDOCOA gene, IDEX4, is increased in the early developmental ventricular conduction system, and then while they are maturing, is almost turned off. Means they actually expression pattern is almost opposing an antagonistic level. Then what is the role of IDEX4? So which is completely unknown. So Rimsha look at their function of IDEX4 knockout to examine the role of IDEX4, and Rimsha and the Jana found that in P21 and P56, so 8 weeks old heart showing the greater prolonged QRS interval, which is the ventricular activation is prolonged. So as you can see here. And she also looking at the conduction system structure and compared to wild type, which is, I'm not sure you can see very clearly, that is like very well developed mesh type of network. The ventricular IDEX4 knockout has some abnormal structure. So she quantified it quantify the data, the images, and then fiber percentage area is significantly reduced in IREX4 knockout. And also the fiber angle is different. So unlike the wild type, which is a blue here, they're more like a diverse direction of the angles. The IREX4 knockout, the angle is more like a vertical, not in a lateral. So this creates some of the functional changes in the ventricular conduction system. Interestingly, when we're looking at the IREX3 and IREX4 level, the IREX3 expression is increased in IREX4 knockout mice in both mRNA and protein level. And if you're looking at the IREX3 expression in a laxity portal mice specifically, so IREX3 is specifically expressed in this Purkinje fiber and the bundle branches in a very specific manner. But when IREX4 is lost, this expression is not specific anymore. The expression, the domain of expression is expanded, suggesting that by losing IREX4, the IREX3 expression is increased. Also, the expressing domain is increased. So it means IREX4 likely restrict the expression of IREX3 by some antagonistic role. So we're looking at the other, the vice versa. So we're looking at the IREX4 expression level in IREX3 knockout. And we found that in the ventricular conduction system specifically, that IREX4 expression is increased in IREX3 level. So it means they likely have antagonistic role in suppressing each other. So the proposed model is, so from the trabecular development to the ventricular conduction system or contractile cardiomyocyte, the balance between IREX3 and 4 decide their fate, cell fate toward the conduction system or the mature cardiomyocyte. So the question is, what if we actually completely remove this balance? So we generate the IREX3 and 4 double knockout mouse and see how the conduction system or structure is developed. And this is the optical tomography data. And so in our control mice, which is double heterozygous mice, and you can see the two papillary muscle in the left ventricle is well developed and as expected. While, as you can see in the double knockout mouse, this papillary muscle is well expanded, it's a hypertrabeculation. Also, this papillary muscle is connected. So as you can see, this is and clearly showing that the abdomen, when IREX3 and 4 is lost, the trabeculation is actually abnormal. So we're looking at the IREX3 knockout and IREX4 knockout, also double knockout. And in the IREX3 knockout, there's relatively normal papillary muscle, while IREX4 knockout, they start showing some non-compaction phenotype. When 3 and 4 is completely lost, they have very disorganized trabeculation, which is compared to the human left ventricular non-compaction phenotype. So trabeculation phenotype and trabeculation is from the embryonic development. And through this early development of the heart from the cardiac crescent to the fetal heart, and there are all trabeculation is completed in the early heart, like E9.5, 10.5, and this trabeculation going through the multiple process of compaction process that they become smooth muscle. So it's very smooth looking muscle. So at 10.5, they are more like kind of protrusion of the cell, and this become shorter and shorter, and they're making a smooth like a compact myocardium. So we're looking at them, different stages of the heart during the development. For example, E14.5 compared to wild type, and which shows a very thick compact myocardium with trabecular region, compared to this control mice. Double knockout mouse has the highly expanded non-compaction region versus smaller, thin compact myocardium, which is also consistently found in at the later stage of the heart, suggesting that IREX3 and 4 plays a critical role in the early development and decide that their fate changes, and then that actually the non-compaction phenotype is derived from the development of the event. So to understand in a molecular level that how this is happening, so we utilize multi-omic sequencing So we got the ventricular nuclei and performed the multiome sequencing to capture both transcript and then the ataxic. So transcript, so we measure the RNA, or the ataxic capture where the DNA is open chromatin, so that gene is regulated. And so there are number of genes, number of cardiomyocytes, and different types of cells are involved, so we can identify them. And as a result, what we found first is this light blue color, the trabecular cell population, is significantly expanded, which is consistent with this non-compaction phenotype in double knockout heart. And but interestingly, when we're looking at this cell in a gene level, the trabecular markers, as like SLE2 and SEMA3A, all these marker gene is significantly downregulated. Means their number is increased, but their property is actually reduced. So they become something not fully well-developed trabecular cell. They become developmentally, I would say, misdeveloped or abnormally developed cells. And why this is happening? So now we are currently working on it. And one of the clues that we found is compared to the non-trabecular cell, so we're looking at the cell cycle of the stage of these cells, and non-trabecular cell, the cell cycle is relatively comparable. But in trabecular cell, if you're looking at the specifically, this S and G2M phase cell is significantly increased compared to control the heart, suggesting that they are actively trying to replicate. But at the same time, we had a very interesting finding that it probably the result is stage dependent manner, because when we're looking at the pH3 staining, which is marked the M phase, the proliferation, this pH3 staining is actually reduced. So suggesting that there's some possible interpretation is they're trying to proliferate, but there are kind of stuck somewhere in the cell cycle, so that they cannot properly differentiate into the mature conduction cell or the contractile cardiomyocyte. So I'm not going to go through in detail, but we also found that using this the method, we identify the number of transcription factors likely working together with IRX34, specifically like a KLF12 and CT12 and E2F8, which are actually the regulator of the cell cycle. So together in this study, we found that the balanced antagonism between IRX3 and 4 require proper the trabecular differentiation and non-compaction cardiomyopathy prevention. And this finding suggests that conduction system development, proper development of conduction system is essential also to prevent this non-compaction cardiomyopathy. So this may explain why the patient with the non-compaction cardiomyopathy often it comes with a conduction defect. So this explains not just the structure, but also maybe the abnormal development of this process. So with that, I'd like to thank all the previous and present the lab members of the lab. And this was a fun part of my journey in the research. And also, I cannot do this also without many collaborators within the institute and also the outside institute, specifically the people who very kindly provide the model system and also share their expertise. And I'd like to also mention Dr. Glenn Tibbett, who's been a great supporter of our research, but who actually unfortunately passed away. So I would like to thank his support as well. So thank you very much for your attention, and I'm happy to take any question from you.
But we're not hiring to the pay. People online watching later and recording. What we're going to do, ask questions, unload them, so that I go into the recording experience. So to start the question, if I may, first of all, that was amazing and wonderfully complex, yet, well, I don't say easy to understand, but very much you really showed us the highlights and most important parts, which I appreciate. I really liked your study when you looked at the convergence divergence across obesity. Not only is it very important, obviously, we learn a lot from conservation, obviously, but these are conserved, and we assume that these are important. But actually, I think what's really interesting is that having something not be conserved and actually something unique to a species might also be able to teach us something like we know embryonic mouse parts can regenerate really, these kinds of things. So that's why I think that's really interesting. I have a question about your overlay with the GWAS and non-limiting variants. So that was striking. There were a number of them that were really aligned with human GWAS for various, conditions. How can you take us to the next level? I mean, how can you look at obviously scales, things like that? You looked at the coding variants, done randomization, looked at sort of those candidates and thought, okay, maybe people have done and knockout. How do you take all of that work that you put in and put it on overlay non-sort of human disease, and now we go to function? I mean, it's a long list. So what do you do next?
Okay, so the question, if I recap, that we identified a number of genes that are divergent and convergence in the cardiac conduction system, how we can utilize that to link to the actual human disease. So it is very difficult job because like how we're going to actually nitpicking, and then we don't have a system. Hopefully, I mean, of course, if we can do, we can make a, like kind of CRISPR based, screening method and then do the IPS system. I mean, if we can have some kind of great way to do so with the funding, I mean, that would be the ideal way. But that's a very time consuming, resource consuming. So we actually have to be a little bit manually picking, hand picking. And indeed, we actually found some transcription factor. I was going to share, but actually decided not to do it today. But we identified some transcription factor, which is completely unknown. But we found that this transcription factor is highly associated with the atrial fibrillation. So, but they are expressed in a SA node specifically. So how is it related? We still do not know. So we did some of the study with Dr. Wenbin Liang, and Jerry also did some of the work together. And we actually overexpress this in cardiomyocyte, the IPS-derived cardiomyocyte, and they become like SA nodal cell. So it means this gene, we had no idea. We overexpress them, they actually becoming more like SA nodal cell, means they can actually play a functional role. How is it linked to the, especially variant is another important question. We still do not know how we can tackle it specifically, but it takes time to understand properly. But yeah,